discuss about Streptococcus identification using the colony morphology, their gram stain appearance and their ability to grow on different media, like the basic media we use in the lab, blood agar, McConkey agar, chocolate agar. So you can see there are differences in different streptococci groups. So how you can use those specific features to identify different groups of streptococci. There are different ways of classification of streptococci. Now you know streptococci means they are gram-positive cocci in chains. We can differentiate them depending on their colony morphology. One thing, their size of the colony, and some are pinpoint colonies, some are little bigger colonies, likewise. Or else depending on the type of hemolysis. On blood agar, you can see in this picture the blood agar. You have seen in the previous slide also how the blood agar plate looks like. Now when you grow the streptococcus on blood agar, different streptococci can cause hemolysis on the blood agar. That is because they can produce different hemolyses. So there are three types of hemolysis. You can see beta hemolysis, alpha hemolysis, and this is non-hemolytic streptococci, or sometimes we call them gamma hemolytic for the non-hemolytic streptococci. Beta hemolysis means by producing the different type of hemolysin, they can completely hemolyze. They can show complete hemolysis on the blood agar. Now in this one, if I show this plate to you, can you see the pinpoint colonies? Because most of the streptococci, they are very small colonies compared to the staphylococcus colonies. And around the pinpoint colonies, you can see a large zone of clearing area. The blood is hemolyzed. Red cells are lines. So you can see through even the hemolysis area. It's transparent. Is it clear? So this type of hemolysis is called beta hemolysis. What are the examples for beta hemolysis for different streptococci? That is group A streptococci, group B streptococci, and C, G streptococci. Those lancetid groups of streptococci A, B, C and G, they all show beta hemolysis around the colonies. Here you can see it's a wide zone of beta hemolysis. Now when you come to the alpha hemolysis, that is partial hemolysis. Not like in the beta hemolysis, the organism cannot hemolyze fully. So there will not be transparent zone around the colonies, but you will see some greenish color, discoloration around the colonies. That's due to the partial hemolysis. So you can't see through the hemolytic area. Again the colonies are very small because it's a streptococcus. Is it clear, the greenish color around the colonies? So the examples for alpha hemolytic streptococci are the Streptococcus pneumoniae and the viridans streptococci. Those are the common examples. Then the other type of streptococcus, depending on the type of hemolysis, is the non-hemolytic streptococci or the gamma hemolytic streptococci. When you look at this plate, you can see again the small colonies. And there is no clearing around the colonies. That means no hemolysis around the colonies. So usually, now this is described as gamma hemolysis, though there is no hemolysis. The examples are group D Streptococci. Group D Streptococci has again another Streptococcus called Enterococcus, or Group D Streptococcus, Streptococcus bovis, like different organisms. And now these organisms, they don't show hemolysis around the colonies. So depending on the type of hemolysis, we can use the property of hemolysis to identify different streptococcus.